University, especially DBT Center. So I will take you to the journey how the biotechnology is depend on the microbes. Okay, everybody, if you look at the images and all those things, how the beautifully these microbes appear in the scanning micrograph. If you look at the word, what they are doing is now everybody is talking about repurposing of bugs, repurposing of drugs. Okay, you take out the corona COVID cases where the drug which has been not meant for the COVID, we are repurposed it, right? But nobody is looking for the whether we need to repurpose the bugs. So this is my hypothesis that we needed to be repurpose the bugs for the biotechnological application. If you look at the geography, 70% of the living protoplasm on this planet is of microbial origin. And without microorganism, you can imagine all life on Earth could have been seized off. And even we know the magnitude of 10 raised to power of 14 to 15 bacterial cells than our own cells. So as I told, without microorganisms, all life on Earth would have been seized. You can imagine any higher animal died in your campus without, in a stipulated timeline, it is not degraded, how the condition in that campus. This is mainly done by these microbes. As I was talking, each and every cycle in the earth is dependent on the microbes, start from the biological fixation, industrial fixation, fertilizer input, the dead organic matter, plant uptake. Each and every cycle is dependent on the microbes. That's why we told 70% of the protoplasm is of microbial origin. However, this is not the only cell numbers are more we have to look for these organisms. They provide the foundation to our biosphere. The frontier of life are also determined the prokaryotic microbes. You look at the people are studying the Mars and all those things, what they are looking. The first priority to look at the whether microbes survive in that environment. So based on that only life will be decided. Right? As you know, uncultured food represents much more than the what we know the culturable one. So we say that only 1% of microbial we know till day, but it has gone say last 10 years, say around 1 to 2 decades. So now it has reached to 5%, but nowhere it has been recorded. So if you look at the biodiversity is a source in biotechnology. Okay. When you call see what is if you look at this is the biodiversity. When any microbiology lab, if I ask they do the isolation, they take a 1 gram of soil, they do the serial dilution, and you'll get millions of microbes. This gives the biodiversity. Then what is biotechnology? It is nothing but art of exploiting the biology. So from this hundreds of microbes, which one you pick? How you grow that culture? So you look at from the hundreds of organisms. So one of my students picked the culture and she grow according to the requirement. This is nothing but this is the Ganesh idol you can see. So this is the art of exploiting biology. So if you look at the development of biotechnology is mainly depend on the identification of microorganisms. Can you imagine the without Taq polymerase, can you run your molecular biology lab? See, this is the fellow who has Dr. Thomas Brook who discovered the Taq polymerase Thermus aquaticus. Till date, there is no alternative for the Taq polymerase. The problem is that we have not looked beyond our territory. So we are a little bit comfort zone that whatever we target, if I ask for any student to look at the amylase screening or protease screening, they grow the media according to the requirement. They see the zone of inhibition or the zone of clearance, they pick that this is a positive for the A, B, C. The rest 99% organism they discard it. So we don't know what kind of activity those organisms are performing. So nowadays, to identify organism, we need a complete information, start from the phylogenetic to the complete genome sequencing. Without that, we cannot predict that what kind of function these organisms are doing. So if we look at the golden era of antibiotic, which we call it as say around 70 to 90, where a lot of new antibiotics have been discovered with a different class. And in last 30 years, there is no new class of antibiotic was being introduced. And whatever existing drugs are there, they are all showing the resistance. The resistance pattern in India is in worse scenario compared to the other worlds. And especially in the COVID, last 3 years, the resistance problem has much more increased than the earlier one. If you look at India is not, as I told, each and every city you take out, you will get a resistance strains in the clinical samples. You look at this is the escape pathogen which we call E. coli, gram-negative, especially gram-negative, see the resistance profile. And what could be the reason? As I told, 30% used by the humans and 70% by the animals. And especially in Indians, we are very, we feel that we are smart. Whatever small fever, cold happens now, we do a self-prescription, which may not be mandated. We take a lot of, go to the pharmacy, we describe to the pharmacist and we take the antibiotics. And if you look at the animals, 70%, because this is the commercial gain is involved. You go to any poultry farm, take a chicken, fly the chicken from 1 meter height. These chickens are unable to open their wings because they poured so much of antibiotic into their system to get the weight. So that's why this 70% is used in the animals. This is the biggest challenge to how to tackle the antibiotic resistance. You may tackle the COVID in a year or two, but the targeting the the AMR is the biggest challenge, ever challenge than the COVID scenario. Then how to measure and where we look for these new organisms. So we call India as one of the mega diversity hotspot. If you look at the microbial diversity study, we are very countable numbers or countable peoples are working on the systematics. The simplest measure of the diversity is the species richness. If anybody have done the sampling in your campus, say around 2010, and the same location you do the sampling 2020 or 22, and you do the how much richness was there in 2010 and how much richness is there in the present, you can see that this is based on this people are predicting the environmental changes also. And whatever the new technologies are being developed based on the diverse organism, take it from the diagnostic, biosensors or biocatalyst. So much more need to be done to understand these microorganisms, invent their diversity, maintain their references cultures, because these references are very much important for the future generation. See, if you look at the coronavirus, we have 5,000 varieties in the family of that coronavirus. Only one has been explored to the animals to the human. How much struggle we have faced in the initial 1 or 2 phase to isolate those, because we don't have any reference cultures. So reference cultures are very much useful for the future pandemics or endemics to look at how to develop how to tackle those kind of problems. As I told, the 80s, 90s as a golden era of antibiotics, what they have screened in that period, you look at the 10⁻³ actinomycetes, which are the predominant group of the organism which produces the majority of the antibiotics. 10⁻³ has been screened for the actinomycin, 10⁻⁴ for chloramphenicol, 10⁵ for vancomycin, 6 for erythromycin, 7 for daptomycin. Any microbiology lab you take it, whenever you do the serial dilution, we stop at 10⁻⁷ only. We never look beyond 10 raised to 8, 10 raised to 9, 10 raised to 10 because we feel that the microbes will not survive in that much of diluted environment. Because now the organisms are found in any environment, we have to extend our screening strategy, looking beyond the 10 raised to 8 or novel or unusual isolates. And because if you look at the historical approach, how they screen for a new lead, they start with the isolation, they do the extraction, then target, then assay develop, then hits. The majority will end up with the new lead. They won't get the new leads because here they look at the strain and the process is very lengthy. The moment you structurally elucidate it, you will end up with the non-structure. Because of the genome mining in last 1 decade, the process has been fastened and rapid way. So high throughput screening strategies have been applied because once you have a genome, because if you take out in 2 decade back, the genome sequencing is not a cup of tea for any research labs because this is the way the cost involved. Nowadays, in 15 to 20,000 rupees, you can generate a genome sequence. Even nanopore, the newest technology in the genome sequencing, which is smaller than your mobile, which can generate a lot of information. So because of genome sequencing, the screening strategy has become high throughput. The based on the once you do the genome, you'll come to know the what are the functional genes. A lot of online tools are there to predict the functional genes. Then you cultivate, manipulate based on the functional genes, then go for the profiling. Which see if you look at the 10 stage process has been reduced to hardly 2 to 3 stages. You look at how the world is looking for the microbes for their various application or molecules. See, grassland, see, India is so rich in the diversity, but we are not exploring that extent. See, people are exploring grassland, coral reef, leaf cutter ants, bee walls. We look at the organisms producing the totally new molecules which have the activity against phytopathogens, indolactic acid, from the actinoplane, which promotes the plant growth. Even some of the molecules, still ecological function has been not known. And the other approach is to first you identify the new organism, look their metabolites. You can see that how they have prepared frulimycin from actinoplane fruliensis, decaplanin from another new species of amylotopsis. So now the molecules are coming out from the new source of the organisms. As I was telling, the abundance, you look at the 1 billion microbial cells per liter of seawater. In the soil, there are more microbes in a gram of soil than there are humans alive today. This is a very interesting study. It has done in 2010 by some of the Western countries involved, around 10 to 15 countries involved. They have dissected the complete microbial flora of the human body. Each and every part of the human body has been detected for this microbial load. So based on this, days are not far away to see that what is lost in the human body can be transplanted through the fecal and all those things. So based on this, you can see how the people are studying. These are the very highly reputed journals where the people are publishing very good. See, gut microbe can help to shed the weight, fight the obesity, antibody treated. Even you can see liver failure can be treated with the fecal microbiota. Stool transplant offer hope to treat alcoholic hepatitis. Gut bacteria boost cancer therapy. Now everything is moving around the microbes. So based on that, we feel that days are not far away. Your pharmacy will be replaced with these synthetic
Our dear participants and students, please give some time to Dr. Syed Dastager. He is trying to reconnect again. So please give some time.
Hello.
Yeah. Thank you, Dr. Syed.
Yeah, I will.
Yeah, yeah. Yeah, you can continue.
Can you see the slides? Yeah, sorry for that network issue. See how much is the online virtual is bad. You can see that one. The physical is the most appropriate things. Okay, so I was here based on the microbial analysis of the human body. So days are not far away to see that our pharmacy will be replaced with the synthetic one replaced with the microbial origin. So based on that, we say that repurposing is needed for the beneficial bugs, not for the drugs. If we look at the extinction, the 150 to 200 species of plant extincts every day, and we don't know the plants. We can visually see that one, but the microbes we don't have any information. The C section, which is 5% in 1970, now it is 30% today, which resulting the newborns are not getting the healthy microbial flora from the mother. Because of that, the lot of infections are happening. Another case, if you look at the skin infection, you people are very fancy. Everybody is fancy to go with the film star advertising a soap. So we see our skin is a very light, slightly alkaline, not the complete alkaline. It is basic to the alkaline one, but we apply a lot of alkaline soaps, which are say around 8, 9 pH, which are removing the healthy flora from the skin. So when we call the skin is the first defense for any infection. When the defense is not there, automatically your skin infections are bound to happen. Things happening in the world. And as I told, to explore the organisms to biotechnological application, you should know that what kind of system it has. This is the very relevant information. If you look at you can see the car, truck, SUV, van, how they have designed, what is their mechanism. If you look at the how based on what they have identified, engine size, mileage, number of passengers, type of transmission it carries, and based on that nomenclature has been given. Tata, Hyundai, Honda City. The similar relevant meaningful system exists in the microbial world, where we need to know the each and every component of the organisms. Without that, you cannot explore the organism for the any applications. So what are those things? You might are aware of that. What level you need to explore that organism. There are different techniques are available. The family level, genus level, species level, strain level. The, so we call the DNA sequencing is the gold standard to know from family to the strain level. As I was talking, can you imagine without E. coli, genetic engineering or molecular biology today? The E. coli is discovered in 1885 till say up to 2008, quite old data, where they have 368,000 publication only on E. coli. You can imagine that we never explore the other organisms for the genetic engineering aspects. So as I told, we are comfort with the E. coli. Okay, we know the system, so that's why we do the cloning and expression happened in the E. coli only. So background, I started my PhD work in way back 2002 to 2006, where you can see that this is the one of the location we Gulbarga, where the soil has the different colorations. The red soil, very black soil, and brownish color. And this region is very known for this limestone quarries. When when the temperature in the winter goes to 5 degree and the summer it goes to 47, sometimes it reaches the 50 degree also. So our aim is to, is there any microbes playing the pigmentation of the soil? So we started looking for the pigmented microorganisms. We designed our protocols. You see that we did the serial dilation. We isolated very unique pigmented organisms. You can see we never see this kind of microbes or exist. Dark blue color, sky blue color, red, pink color, brownish color. So based on that, we did the complete polyphasic approach to understand the taxonomy of those organisms. Start from scanning, electron micrography, then chemotaxonomy. These are very important parameters to understand the microbe cell wall. Fatty acids, polar lipid, then whole cell protein. Then we did the sequencing. Based on that complete analysis, we came across with the first new species from the Gulbarga. So we proposed the Streptomyces gulbargensis. As I told, whenever you want to nomenclature any microbes, there are certain set of guidelines by the International Union of Systematic Microbiology where we propose the names. So based on that approval, it will be recommended. There are three categories which usually taxonomists follow. One is the geography from where it has been isolated, the property of the culture which it harbors, and the second where the taxonomy has been performed. Some of the labs also been proposed, which you can witness in the next slides. And very interesting aspect, whenever people have any new organism, when they want to compare with the phylogenetically closest strains, they compare the data from the published information. Either they refer the Burgess manual or in the internet. That is not the right way to compare. When you come to the see, my strain is very close to the Venezuela, I cannot take the data from the published information. We have to perform these parameters in a given lab conditions. You have to procure the tribe strains which are very close to your isolate, then you mimic all the conditions what you are analyzing. It's a valid comparison. Otherwise, this is not considered for the valid comparison. Similarly, we propose around 4 new species from, as I told, tri-tolerance. It is tolerating to pH, temperature, and salt. Based on this, it has proposed the tri-tolerance. Halophilus, it loves to grow in a saline condition. Same Saccharomyces salifila. Streptomyces deccanensis. Deccanensis with the Gulbarga region comes under Deccan Plateau, where we propose as a deccanensis. As I told, the taxonomist has the privilege to give the name, how the parents have the privilege to give the new names to the babies. The similar way, the taxonomists have the privilege to propose the names. Based on this will be considered. So once we have a new strains, what is next? That too, when you have a streptomyces or actinomyces in your collection or in your lab, the first priority comes in mind that we should look for the antimicrobial activity. The similar thing will happen with us also. We started with screening for the actinomyces for a secondary metabolite, especially antimicrobial activity. None of these organisms shows any antimicrobial activity. So we thought, what makes this organism different than the other organisms? When the whole actinomyces are known for the secondary metabolites, then we look for the different enzymes, whether these organism produces enzymes Very surprisingly, you can see that. So this is the initially we studied the pigmentation. Now look at this one stage. Streptomyces pulverulens shows the very potential enzymatic activity for the protease, amylase, and even keratinase. You can see the one organism playing how the activity changes. It demonstrated almost even chicken feather degraded within 96 hours by this organism. So where I completed based on this, I submitted thesis. Then I went for a postdoc in Korea Research Institute of Bioscience Biotechnology. Basic foundation you got it. What you have to look for. So based on that, we did the survey of the geography from the lab. What is the what you call uncontaminated area to look for the newer organism. So there is a Bigam Island which is in Japanese Sea, which has been untouched land. We went with the one of my Korean colleague. We together went. These are the only two alien in this island. Nobody is there. Unmanned land. We did the sampling there. We came back to the lab. We analyzed those things. We did the systematic approach to reveal their taxonomic identity. We reported around 16 new species from one locations. You look at this. You look at this red font one, the nocardiates dilutes. So when you make a nutrient agar, it is a 100 strength. We call it as a 100 strength agar. This media has been diluted 100 times because that the island, what we assume that island doesn't have any nutrients. So based on that, we mimic the condition. Still, the organism grows in 100 times diluted nutrient agar. Another interesting organism is Agriphilus. You don't need to put any carbon or nitrogen source. Simply put a 2% agar, this organism happily growing in the agar. So based on this, we have proposed so many new species, but still this institute is exploring this organism for the various applications. So then I moved to the National Institute of Oceanography, where I worked on the marine. As I'm touching all the aspects of the soil, island, and marine, which gives this. Once you have a basic foundation in microbiology, you can explore whatever to do. Whatever lab or whatever lab you choose. Here also we did the identify island, which is again unmanned island near the Goa. We did lot of isolation strategy involved in this one. We did the isolated the different isolates. You see the how beautifully they arranged in the scanning electron microscopy. And we proposed in last 5 to 6 years, we proposed around 27 new species, including 2 new genera. So we have around 60 to 65 new species in our collection. Now comes what is how to explore those organisms. We are fortunate to have our genome sequencing facility in our collection. We started the complete mining these new organisms. We want to know that based on the genome mining, how we can explore these organisms. So we targeted certain metabolites like ladderin, which are highlighted in yellow font. You can see that these are the very unique metabolites which have been not reported from the respective organisms earlier. So once we know the functional genes, we mimic those conditions, we manipulate the experiments, and we analyze those activity profiling. And this is the one such example. As I was telling, this is the Arthrobacter. You read the NCL and CC. NCL is the National Chemical Laboratory. E is silent. Here you can propose as the NCL and CC. So Arthrobacter is very well known to degrade the pH, polyhydroxy butyrate molecules. But till date, Arthrobacter is not known for the antimicrobial or bioactive potential, secondary metabolite. We analyzed the 20 genomes we compared with the first Arthrobacter strain has the PKS1 molecule. which polyketed synthase, which indirectly correlate to the secondary metabolism. We analyzed by the PCR to demonstrate that the PKS gene is amplified in this DNA sample. So, as I told, there are two arthrobacteria have been isolated from the same location. We did the antimicrobial profiling of those things, resistance profile. See that the organism which has its own system PKS is showing the resistance because it has its own antimicrobial potential. The another organism which doesn't have the antimicrobial or PKS producing ability, it is showing the sensitivity. Based on this, one of my students has explored this organism for the acarabose production, which is the alpha glucosidase inhibitor for the diabetic potential. We procured the actinoplane, which is the engineer organism which is known to produce the actinoplane, acarabose. And the standard acarabose also we procured, we compared all the properties. You see that this is the actinoplane, which is engineered strain, which is commercially used for the acarabose production, and this is the arthrobacter. You can see this is the standard acarabose. You see this is the pink color pattern, which is totally different than the actinoplane, which is a very model organism. So we are purifying property. We did a lot of analysis. We are working on, still work is going on this aspect. So based on this, first time we reported that arthrobacter also has the secondary metabolism for the activities. Another example is the Rhodococcus encielensis, and shows the functional genes of phenazine. The phenazine is an antifungal compound predominantly produced by the Pseudomonas. So none of the Rhodococcus till date exist has shown the phenazine. So once we know the phenazine, we did the antifungal potential. It is a time-lapse scan micrography we did. You can see that when you make the co-culturing of this Rhodococcus with the fungal pathogen, you can see how it start inhibiting the fungal mycelia. You can see these are the Rhodococcus attacking the mycelia. You can see here completely attacked. You can see that they start breaking the mycelia of the even if you see the fungal spores, how they disturb their structures. So this is a very interesting observation we come across. Whenever any secondary metabolite work is going on, people will stick to one or two medias. Secondary metabolite media. Here also we did initially we started with one organism with one media. We end up with that there is no activity. When I went for a Germany for a training under Raman Fellowship, I came to know that whenever you have a set of different media, at least 5 to 10 media, then only you will come to know that which media is supporting the secondary metabolism. If I go stick with the one media, I will end up with there is no activity. When I put the same organism in a different media, you can see the activity completely changes. You see that 5 for some media where the activity is almost say 90% activity has been demonstrated. In culture, if I go with the standard media which has been referred in the maximum literature, I will end up with there is no activity in that one. See, this is a very interesting aspect there. Whenever secondary metabolite screening happens, you should stick to minimum 10 media, different metabolite production media. Because in this case, carbon source are very much important for the metabolite secretion. Based on this, you can see that how this time-lapse MIC we did for the fungal. You can see the micromolars, 0.53 millimolar. It is inhibiting complete the spore formation. These are some of the examples where we have reported the new organism for the Mycobacterium, the chrysomycin from the Streptomyces species against Mycobacterium. Till date, chrysomycin is not known for the MTB activity. Similarly, another metabolite in cervical cancer cell lines, and this is the rapamycin is the only known molecule which inhibits the mTOR pathway, and that molecule inhibits partial pathway. But this metabolite completely inhibits the pathway. So it published as a two extract inhibiting. Later on, it took 4 years to elucidate that structure. We did a lot of analysis with the NMR because since I'm in a chemistry lab, we see until unless you do the interdisciplinary research, you cannot explore your microbes for the any extent. Biology, pharmacology, all background should be there to explore any new molecules. We did the NMR structure. These are all the profiles of those molecules. Then we did the mass spectroscopy to understand what is the mass of that molecule. And as I told, this is the structure elucidated. You can see that chemically it cannot be synthesized. These are microbes are so fortunate have this kind of structure. It is very difficult to synthesize these molecules. So you should look at the organism which are producing very unique. Based on this, we have recently demonstrated it is angiocycline like compound which inhibiting the both pathway. This is the first biological molecule which is inhibiting the both pathway in the cervical cancers. This has been published as a cover article in the ACS Clinical Biology. So later on, another one molecule which my lab is working since last 5 years on a nigricin, which is reported from a Streptomyces hygroscopicus, and it was isolated in 1950, and it has a broad spectrum activity against gram-positive fungi, tumor cell lines, including HIV, viruses, and HIV. These are the literature supports this molecule has been explored for different application. One of my PhD student has isolated from the dead plant. This strain has been isolated and it is not related to hygroscopic. There is no close relation of hydro. This is totally different organism. We did the 50 liter straight away. Once we know the activity, we go for 50 liter fermentation. We extract the 75 gram from the 50 liter fermentation. We did the all NMR structure to elucidate the structure of this molecule. Standard device is available in cinema. So 1 mg is costing around 1700 rupees and it is only available for the research purpose. We compared with the standard, a lot of crystallography to predict this their crystalline nature. So very interesting, this molecule has shown the anti-malarial activity at the picomole. You can see that it is showing the 0.980 nanomole. It is almost 900 picomole. As I told, this molecule has been not come out to the are taken as a candidate because of the toxicity. So we took a challenge in that one. When the people have explored this organism in 50s, 60s, that time they don't have the toxicity profiling or experiment so sophisticated instrument to do the toxicity analysis. We took a challenge to analyze complete toxicity. We spent around 20-25 lakh rupees to generate this toxicity profile, outsourced with some industry. You can see that we did the complete plasma concentration, toxicity profile, both male and female, and we did the histopathology, and even we did the 14 days repeated study to see that how this affects the humans. And based on our study, we came to know that after 14 days repeated intraperitoneal route, this molecule doesn't show any adverse effect on the rat. So this gives something hope to take this molecule for further. We take this molecule for anti-cancer activity in Akshak, Mumbai, where it has demonstrated this molecule inhibit the almost all cancer cell lines available in the Akshak center. There are around 25 cell lines of cancer. This thing they have shown that this molecule is inhibiting almost all cell lines. And we took the two genograph model to see that how it works. The one is the colon cancer. You can see that even they used the adriamycin, which is a standard drug used in the chemotherapy. At 1 mg concentration, this animal died with the standard drug. Has been been observed, and you can see the controlled tumor and you can see the reduced tumor within a 1 month. So you can see there is no weight loss, nothing has been observed. You can see the animal body weight has retained as it is for 30 days. Repeated dose. As I was talking of survival data, when the standard drug adriamycin, which is already used for chemotherapy, when you give for a repeated dose, you can see one animal died at 12 and another at 15. But nigrosin animal never died in 30 days of repeated dose. Similarly, we look at the liver, lung cancer cell lines. You can see similar observation was made in the given adriamycin. Again, it has shown the lethal dose at 0.5 mg, 1 mg, and 2 mg. And this molecule doesn't show any lethal dose up to 30 days. So this is the very based on that when we are literature searching that what make this new review came in 2021 where they show that evidence of nigrosin as a potential therapeutic candidate for the cancers. Whatever we analyze, this is giving us some confidence that this molecule can be taken further. Where you see we mentioned very clearly that to increase their bioavailability, they have to find out the new administrating drugs, a new system for administrating this drug, and the route increase the bioavailability, and improve their therapeutic efficacy. So based on that, since as I told, we are fortunate to have a good chemist with us. We modified this nigrosin into fluorinated one. We derivatized this molecule, adding the fluorine to this one, and very interestingly, this nigrosin is till date known to inhibit only gram-positive organism. When you modify this nigrosin chemically, it starts showing activity against gram-negative organism. You can see that it is showing very prominent activity against gram-negative organisms. As I told, the toxicity, it has 10 times toxicity has been reduced from the parent molecule to the modified version. If you look at the parent molecule is 10 micromole is the toxic, the nigrosin modified nigrosin 400 micromole means it has reduced 10 times in its toxicity nature. Why we are looking for the fluorinated? If you look at in 2021, FDA approved so many fluorinated molecules. Even you can see 25% have been launched. Many are in phase 1, phase 2, phase 3 or pre-registration. So based on this, this has given a very boosting information for us to fluorinated can enhance the therapeutic efficacy. It gives the new route. And we have signed an agreement with the dental, medical college in Harvard. They are exploring this molecule for the AMR strains. And very interestingly, in 2020 or 21, we give this molecule to NIV to testing for the SARS-CoV also. It has shown the very good promising activity. Still NIV is studying the animal study of this molecule. And even it has shown the inhibition for the Delta variant also. So we analyze with the comparison with the nigrosin and remdesivir, how it is different than the remdesivir. It has very potential than the remdesivir. So as I told, whenever you look at the molecule, we have to look the cost calculation is also very much important. As I told, 1 mg of the sigma molecule, the same nigrosin, 1 mg is costing around 1700 rupees. When you can, microbes have the capacity to produce the molecule in 1 gram at 600 rupees. We demonstrated that we can produce the molecule in 1 gram at 600 rupees. This is another interesting molecule, indolmycin, which we are working on it. Again, we did a lot of structural elucidation. This molecule is known to only have a staphylococcus, anti-staphylococcus activity till date. In our case, it has shown the activity against MDR tuberculosis. You can look at the multi-drug resistant tuberculosis at MIC 20 mg per ml. So we are working on those aspects. And this is another, this is some of the examples what we are exploring the microbes for the various biotechnological application. So this is the bacterial cellulose. Whenever you talk about cellulose, cellulose majorly comes from the plant origin. So there are the organism which produces the cellulose. We have isolated a very unique organism which produces the bacterial cellulose. We demonstrated at a 50 kg scale. This cellulose has a vast application. Majority of the biomedical sector, membrane, food sector, pharma sector, we are exploring different application of this organisms. So in COVID also we tried bacterial cellulose. It can be coated on the medical grade cloth. We demonstrated that it is showing the 99.9% bacterial filtration efficiency. This is much more better than the N95. We transferred the technology to a local MSME during the first phase of COVID. So when the N95 that time they are selling around 350 rupees. We demonstrated this one can be produced at 35 rupees N95. So at that time we have supplied around 20 lakh masks to the Maharashtra government. So by using bacterial cellulose, we demonstrated certain chemistry experiment as we told you. Reaction can be enhanced by using the bacterial cellulose support system. This is the one of the drawback in the chemistry where they use the TBAF as a catalyst, which is a hygroscopic nature. Within you can see that within 15 minutes this will become liquid. By using our material, we demonstrated that reaction can go up to 21 days. Another molecule which my student has worked on the biosurfactant from the marine organism, we demonstrated this has a very good potential for the oil remediation. You can look at how beautiful this the oil from the water. This is the crude what do you call it, garage oil mixed with the water. So we use this supernatant of that biosurfactant, which is produced in the 11th hour. Whenever we did the initial isolation of this molecule, we are getting very difficult time. We go for 5-day fermentation. We are unable to purify this molecule. So I told a student to stay in the lab. Every hour you do the sampling. We came to know that the molecule is produced at the 11th hour only. We harvested from the 11th hour, then structurally elucidated. See how so you can look at how microbes are producing this kind of complex molecule. The structure when we analyze it is C95H190, which is very unimaginable from the microbes. Why they produce this kind of complex structure? And another interesting aspect of this media is just a tap water and yeast extract. You don't need to put any carbon source, nothing. Just put a tap water and yeast extract. Here I am putting a supernatant of that fermented broth on a normal filter paper, just 1 ml. Within 8 seconds, it separates the oil from the water. So this is the one of the very good. So we are working with some of the industry on the bioremediation or oil recovery kind of thing. So you can see this within 8 seconds, it separates the oil from the water. So when we analyze this water, no, there is no traces of any oil in that water. It completely removes the oil. So whenever you do any good science, you need to have a backbone. Without that backbone, you cannot do any good science. So here I consider the backbone is the human resource. You should have a good student to execute your ideas or your work. So these are the gems of my lab where they work on different aspects. Phylogenetically, in that one. And 5 students have been awarded PhD. Some are already settled in the industry and all. The second aspect for the backbone is the money. If you have a good student, if you don't have money for a consumable, how you run the lab? So these are the two parallel lines go hand by hand. One, you should have a good human resource. One should have a good funding for the lab. These are the projects we executed in last 5 years. Some are in pipeline. We got this year. We got around 4 projects from the DBT. We are executing all those. So this is one of the unique culture collection. It exists at NCL. This is started in 1950. These are the mother of all the culture collections available in India, including IMTech, Chandigarh, NCCS, and MAU, the agriculture important microorganism, which is in the NBIM. So all are generated from this culture collection. This is the very unique culture collection which is holding 5,000 industrial microorganisms. We serve the cater to all the industry research organizations throughout the country. Have a state of art facility from the starting with isolation characterization to the production characterization of the molecules. We say that we have a two doors, enter with the culture, exit with the product. We have complete facility here. And this is one of the highest revenue generating unit in the whole country in the science and technology forum. So we have two scientists, five technical staff, we generate 5 crore lab reserve per year. Of COVID also we generated last 3 years we generated around 5 crore every year. So this is better and better of our unit, and 30% of what we generate will come back to the lab to maintain the facility and upgrade the facility. So these are my research lab where I started my learning with Gulbarga University with Professor Dan, and now he's the Vice Chancellor of that university. Then I went with the Yunnan Institute of Microbiology to learn the systematic with one of the renowned taxonomist, Professor Wenjun Lee. Then I was in the Korea with the postdoc. And these are my labs where I started my earning. These are the learning labs and these are my earning labs. Thank you.
Yes, it's open for discussion. Students, if you have any questions, please ask. Possibly they have understood everything, Dr. Syed. Yeah. Or probably they are thinking about their own research, that they can plan it like this and they can do it like this. Sure. Yeah, most of the students, they are interested in microbiology.
Okay.
I've been talking to them and I think they are interested in microbiology. Yes, anybody has any questions, please? Also, online participants, if you have any questions. Students, you have to come forward a little bit and ask. You don't have anything else? No? No questions? Yeah, I think you guys are. What else? Okay, I think they don't have any questions, sir.
Yeah, fine. No issue. Yeah. Thank you.
Thank you so much. Thank you so much for your presentation.
Yeah, thank you.
ऐसी के तो अपन ऑक्सफोर्ड कॉलेज में वो सुनना बस You take a break? No.
I think they need a break.
Huh?
They need to.
10 minutes break. हाँ जी, very critical. Morning time is I say it is no problem. Meeting at house. अरे 1:30 का probably सोचते हैं 2:30.
One minute.
More. कभी कभी जब हम फ्री होना है. वो आके नहीं जब वो फाइन है तो मतलब फ्री हो गए.